Alright, welcome back to the third part of macromolecules, and in this one we're going to talk about proteins. I'm going to try and keep this as brief as possible because proteins are so important. I mean, carbohydrates give you the ability to produce ATP for energy in your cells, but technically you could live without them. It's the only macromolecule you could. Fats, incredibly important for structure in the cells. Without phospholipids you'd have no cell membranes, you'd have no organelle membranes, everything would fall apart, your cells would disintegrate. Cholesterol, again, gives you all your signal messengers and your endocrine system, as well as holding those cell membranes together. You still get ATP from putting triglycerides into the respiration pathway, you get protection, etc., etc. And nucleic acids, we're going to talk about next, is the fundamental part of our building blocks of our life. In all our nuclei, we have our DNA, which, are sequences of DNA make up genes which code for proteins. The only thing our genes code for is the macromolecule protein. So we immediately need to understand that because that's the only thing that we pass on to the next generation, proteins are incredibly important, not just for life, but to determine how that life develops and proceeds. So to try and really hit the cliff notes of this, I just want to focus on the main things. So proteins, the only molecule which is coded for by our genes. What are they made of? Well, like carbohydrates and the fats, they have carbon, hydrogen and oxygen, but they also have nitrogen. So they have a new one, nitrogen. So, of these, etc., they have monomers, and the monomers are what we call amino acids. And they are the building blocks of proteins, and they're also essentially the way it will be determined what shape protein has. Now, an amino acid has 4 parts to it. You've got a central carbon here, and then above it you've got a hydrogen, and below it you have a variable group, R. And this is what determines what type of amino acid you have. So some can be as simple as just a hydrogen there, or one more carbon and a methane group or something of that nature, but some can be very, very long. On the left hand side of the carbon you've got an amine group, which is a nitrogen and 2 hydrogens. Nitrogens can make 3 bonds. So 2 hydrogens and then bonded to the carbon. On the right hand side you've got an acidic group. An acidic group is COOH. The amine group is NH2. And hence you get the name amino from amine, acid from the acid group. And these are always the same. It's the only thing that changes from one amino acid to another is this part here, the variable group. And so the monomers join together through peptide bonds. And a peptide bond takes off the hydroxyl here, and it takes off a hydrogen of the next amino acid next to it. And there's your water. So when you take that water molecule out, you've still got, I'll redraw this slightly, you've got your carbon there, and then you've got your nitrogen with 1 hydrogen bond there. There's the second, there's the third. Carbon makes 4, 1, 2, 3, 4. So there's your peptide bond between a carbon and a nitrogen. And peptide is what you call a short protein. It's basically called a peptide because it's made up of peptide bonds between the carbon on the C terminus and then with the next amino acid, the N terminus. So the variable group determines whether the protein will be water soluble or not. And that's because in the rest of this you've only really got the 1 oxygen up here when you've actually joined it to something else. So what happens then, if it's just a hydrogen, this isn't going to be water soluble. It doesn't have enough charge on the molecule. But if there's a long chain with lots of oxygens on it, then it is going to be water soluble. Of all of the amino acids that we know, about half are water soluble and about half are not. And that's important because proteins work in the fatty membranes as well as they work in water. What do proteins do? Well, they have so many functions. I've only listed a few. This is not an exhaustive list. Then we're going to look at transport next, and so we're going to see how they help things move across the membranes of cells, how they move in blood, how they move in liquid water exactly. Then once something's actually inside the cell and we want to do the chemistry to keep the cell alive, well, the proteins can act as enzymes, and they can actually create a reaction where normally it wouldn't occur. So they facilitate reactions. They can act as messengers going from one cell to another or even within a cell. And they are a major part of the structure of the human body. Bones are made up of tightly packed collagen. Collagen is protein. And I could go on and on about all the various different structural aspects. Every cell's held together with microtubules and microfibers and intermediate fibers, and they're all proteins, etc., etc. The nails are proteins. Our skin's full of keratin, which is a protein. So, structure, and then receptors. If you let go of a messenger and it can't go straight into the cell like a steroid could, it's got to bound onto something. Well, that's a receptor. That's also a protein. So because that's all we code for, and they have so many functions, proteins' structure is absolutely critical to their job. Basically, you can switch a protein on or switch a protein off like a light switch. And you do it the same as a light switch by changing the structure. If you think about a light switch just for a second, we all know there's a little plastic plate on the wall, and if the switch is pointing up, like that, say it's in the on position, but then if you flip it, it's now in the off position. Take that off again. So by flipping the switch up and down, you switch the light on, you switch the light off. A protein has a three-dimensional structure, and I'm drawing on a two-dimensional board, so it's not going to make a lot of sense. But maybe it looks something like that. Completely random. However, on that, if it's a receptor, for example, there may be a binding site here and a binding site here. And that perfectly fits another molecule. The distance between here and here is just right for one particular molecule to fit into it. And when it does that, it can break the thing apart and you can get different things coming off it. So it's acting as an enzyme in that case. Now, because of its shape, if you change the shape, and we'll talk about that in a second, then this could now close in. So your binding site is now here and here. So that molecule can no longer be processed because the protein has changed its shape. Alright, so why would it do that? Well, protein structure is based on 4 levels. As you add one amino acid to another, you have your primary level, and that's the sequence, I write them as alphas, sequence of amino acids. And it's because of these R groups. If you've got a whole sequence of amino acids, I'm just going to do them like that, and there are groups coming off. Say this one has lots and lots and lots of positive charge. I really drew that badly. Let's try that one again. Positive, 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 positive. And this one is also positive. Well, you know from magnetism that these two amino acids are going to push away. So instead of being in a straight line, that is instead going to look like that, because this amino acid here is going to come over, R group's going to come over here, and that one's going to come over here. Now if this part here, the next R group in the thing, is all negative, then that one's going to move even further toward the negative R group. So that's going to exaggerate that curve even more, and say the next one on the line is got no charge at all. And so that's going to push away again, and so the line, say, may go up like that. So as you add amino acids along your sequence, depending on whether they have charge and what type of overall charge that they have, they're going to interact differently with the amino acids close to them. And don't forget, these proteins are hundreds and hundreds of amino acids long. So whilst you've got your original line, which is kind of doing that, it may end up bending back on itself. So the R group coming off that amino acid and the R group coming off that amino acid can interact. So what you tend to find is you start off with your sequence of amino acids, and the R groups, if they're charged, may interact through ionic bonding. They could interact through covalent bonding as well. We're not going to get into the detail of how they do this because it's not our function in this class. However, let us just for a minute think about it. So the primary sequence is all down to the R groups, the bits that hang off the bottom end of each amino acid, and they can attract or push away from each other, creating different turns and twists as the amino acid chain grows, as the peptide gets longer. That allows for a second type of interaction, which is the secondary level, and that's the production of hydrogen bonds in between different parts of the protein, because if you look at this thing here, there's lots of parts where they're interacting, and you can make weak hydrogen bonds, and some of these can form as an alpha helix, and some can form as beta pleated sheets. Again, you don't have to worry too much about that, but they're hydrogen bonds that are easily disrupted. The third or tertiary level is the complete protein, and that includes all the interactions of the amino acid R groups and all of the hydrogen bonds, plus any new bonding like disulfide bridge bonds, different types of covalents, the hydrogens, the ionics, the whole thing that's creating that structure. And finally, at a quaternary level, or fourth stage level, if you've got more than one protein working together to do a function, then that's your quaternary. So like hemoglobin has 4 heme proteins surrounding, with iron being held together. Alright, so that's essentially what I wanted to say about. There's lots of other things I could say about the proteins, but this is really the part that I'm trying to get home. The stages, the structure, how that is held together simply by peptide bonds, the monomers, amino acids. I mean, when we talk about DNA later, we're going to talk about these amino acids again and how they translate the DNA code to amino acid code, and that can be read and proteins can be made. But that's for a few weeks hence. I think that will do for now. If you have any questions, please let me know.